The answer is yes, and it goes back much further than you might expect. Ancient humans got sexually transmitted diseases. The story of where those diseases came from is stranger and older than most people realize. Some of the pathogens we treat in clinics today have been riding along with the human lineage for millions of years. A few of them were picked up from other animals at moments of contact that we can barely reconstruct. Some were passed between different species of humans during the same encounters that produced hybrid children, and at least one family of infections may have been living inside hominins long before Homo sapiens existed as a species. This isn't really a story about ancient people making bad decisions. It's a story about what happens when living bodies share space, and sometimes much more than that. Start with herpes, because herpes is where the timeline gets genuinely uncomfortable. There are two main strains of the herpes simplex virus: HSV1, the one most associated with oral cold sores, and HSV2, the one associated with genital infections. They seem like close relatives, and they are, but their histories are actually quite different. HSV1 has been with the primate line for an extraordinarily long time. Estimates from molecular clock analysis suggest it's been evolving alongside our ancestors for somewhere between 6 and 8 million years. That means HSV1 was already present in the common ancestor we shared with chimpanzees. It didn't jump to us from some outside source. It was already there, embedded in the biology of the line before we even existed. It has been along for the entire ride since. HSV2 is a different story entirely. A 2015 study published in the journal PLOS Pathogens reconstructed the evolutionary history of HSV2 and concluded that it didn't originate in the human line at all. The evidence points to a cross-species transmission event from chimpanzees or a closely related ancestor, likely passing through an intermediate hominin species. The study's best estimate places that transfer at roughly 1.6 million years ago. That is deep in the era of Homo erectus. Something happened between our early ancestors and chimpanzees that allowed the virus to jump hosts, establish itself, eventually become a permanent feature of the human infectious landscape. What exactly that contact looked like is not something we can fully reconstruct, but the genetic signal is hard to argue with. The interesting thing about that HSV2 transmission event is how unusual it is from a virology standpoint. Viruses that jump between species often fail. They infect a new host once or twice and then die out because the new biology doesn't quite fit. For HSV2 to make that jump and persist, it had to find enough compatible biology in early Homo erectus to reproduce, spread, eventually adapt fully to the new host. The fact that it succeeded tells us something about how biologically similar we were to the other great apes at that particular moment in time. Our immune systems, our cell receptors, our basic tissue chemistry were close enough that a chimpanzee virus could find a foothold and never let go. We've been living with it ever since. Now move about 1.7 million years further back, because there's something even older. In 2007, researchers published a study examining pubic lice, that are a parasitic insect that lives exclusively in coarse body hair and spreads almost entirely through prolonged physical contact. Humans and gorillas both carry pubic lice, but the versions that live on each species are genetically distinct enough that they diverged into separate lineages around 3.3 million years ago. That date corresponds to a period when early hominins, likely Australopithecus or something very close to it, were sharing habitat with ancient gorilla ancestors in Africa. The lice couldn't survive without a host, and they couldn't travel through air or water. The only way they could have moved from gorillas to early hominins was through direct, sustained physical contact. The lead researchers on that study pointed out that the lice themselves function as a kind of biological record. They track contact between species in a way that's very difficult to reinterpret or explain away. The gorilla version and the human version diverged at a specific moment in time. That divergence requires an explanation that involves actual bodies being very close together. Whatever that contact was, it left a passenger behind that became a permanent fixture of hominin life for millions of years. By the time anatomically modern humans appeared, pubic lice had already been living on our ancestors for longer than our entire genus has existed. That's a long-running arrangement built on a moment of contact that probably wasn't anyone's plan. HPV is another infection where the timeline extends far past anything resembling modern history. Human papillomavirus is actually a large family of hundreds of distinct strains. Research into ancient HPV suggests that at least some of those strains were present in hominins well before Homo sapiens appeared. The 2018 genomic analysis found evidence that multiple HPV lineages experienced their own cross-species transmission events at different points in hominin evolution. Some strains show patterns of diversification that track closely with human migration out of Africa and subsequent contact with Neanderthals and Denisovans. HPV requires very little in the way of transmission infrastructure. Skin-to-skin contact is sufficient for many strains. For a species that was encountering other humans regularly and occasionally having children with them, keeping those infections separate would have been essentially impossible. The Neanderthal interbreeding question is where the STD story becomes particularly interesting. We know from ancient DNA analysis that modern humans with non-African ancestry carry between 1% and 4% Neanderthal DNA. That DNA got there through actual reproduction, meaning Homo sapiens and Neanderthals had children together during the period of coexistence roughly 50,000 to 40,000 years ago. What doesn't get discussed as often is what else traveled alongside those genetic exchanges. When two populations meet and reproduce, they don't just swap chromosomes, they swap everything, living in and on their bodies. Every pathogen, every parasite, every virus that had adapted to one species was suddenly getting access to a new one, with a new immune system it had never encountered before. A 2016 paper in PLOS Pathogens modeled what the epidemiological consequences of that Neanderthal and Homo sapiens contact period would have looked like. The conclusion was that both species were almost certainly transferring diseases to each other during those encounters, that some modern human infections may carry ancestry that traces back directly to that window of contact. The transfer almost certainly ran in both directions. Homo sapiens may have introduced pathogens that Neanderthal immune systems had never been exposed to. That would have added biological stress to populations that were already declining for other reasons. And Neanderthals likely transferred infections back, some of which embedded themselves in the modern human lineage and are still present today. Those interactions weren't just a genetic curiosity, they were a full biological exchange with lasting consequences. Denisovans add another dimension to this picture. The physical remains are so minimal that reconstructing their biology in any detail is difficult, but the same logic applies. Melanesian populations carry up to 6% Denisovan DNA in their genomes. That represents sustained contact and reproduction between Homo sapiens and Denisovans somewhere in Southeast Asia or Oceania. The exact timing and location of that contact is still being worked out, but wherever it happened, it came with the same infectious implications. Two genetically distinct human populations, close enough to produce offspring together, are certainly close enough to share whatever was circulating in their bodies. Whatever pathogens the Denisovans were carrying had a direct pathway into the modern human genome during those encounters. Syphilis is one of the most debated questions in the history of infectious disease. The argument has been running for more than 500 years. The traditional account holds that syphilis came to Europe from the Americas in 1493, brought back by sailors who had traveled with Columbus. The evidence for this came from a sharp increase in documented European syphilis cases immediately after contact with the New World, and the apparent absence of syphilis symptoms in pre-Columbian European skeletal remains. That framing held up reasonably well for a long time, and it still has defenders. But it's been challenged repeatedly. Recent research has shifted the picture significantly enough that the simple Columbus story no longer holds up cleanly. A 2023 study published in Nature puts serious pressure on the Columbus theory. Researchers examined ancient skeletal remains from sites across Europe, several of which predated the Columbus voyages by decades or centuries. Using high-resolution imaging and chemical analysis, they identified bone lesions consistent with treponemal infections in multiple pre-Columbian European specimens. Treponemal infections are caused by a family of bacteria called Treponema, which includes the pathogen responsible for syphilis. The study stopped short of claiming definitive pre-Columbian syphilis in Europe, but it demonstrated convincingly that some form of treponemal disease was present in European populations before Columbus ever departed Spain. The Treponema family is actually worth understanding in some detail, because syphilis doesn't exist in isolation. Treponema pallidum has 4 closely related subspecies, each causing a different disease. Syphilis is one, yaws is another, a skin infection spread through casual contact that has been documented across Africa, Asia, the Americas for thousands of years. Begel causes lesions in bone and skin. Endemic syphilis, sometimes called non-venereal syphilis, is the fourth. These diseases are so closely related genetically that distinguishing between them in ancient skeletal remains is genuinely difficult. That difficulty is part of why the debate has persisted without resolution for so long. What's clear is that the Treponema family is ancient, and that some of its members have been living in human populations for a very long time, well before 1493. Gonorrhea is a different organism entirely, a bacterium called Neisseria gonorrhoeae. Its history in the human species is also older than most people assume. Molecular dating studies suggest the gonorrhea lineage diverged from its closest relatives somewhere between 100,000 and 180,000 years ago. That places its origin in the period when anatomically modern Homo sapiens was first appearing in Africa. Some estimates push the date earlier still. What that means in practical terms is that gonorrhea has been a human-specific pathogen for essentially the entire existence of our species. It didn't arrive recently from another animal host. It evolved alongside us, adapted specifically to our biology, has been a feature of human life since before our species spread beyond Africa to the rest of the planet. Hepatitis B is another infection that ancient DNA analysis has revealed to be far older than previously assumed. A 2018 study in Nature reconstructed ancient hepatitis B genomes from skeletal remains across Europe and Asia, spanning the last 7,000 years. The results showed that the diversity of hepatitis B strains in ancient populations was dramatically higher than what circulates today, suggesting the virus went through multiple rounds of diversification and selective pressure over human history. The study also found hepatitis B in hunter-gatherer populations that predated the Bronze Age, meaning it was present well before the large, dense settlements that are typically assumed to be necessary for infectious disease to sustain itself. Smaller nomadic groups were carrying it too, moving it across landscapes long before anyone built a city. The broader scientific field that makes all of this possible is called paleomicrobiology. It has developed rapidly over the last 2 decades. Scientists can now extract and sequence pathogen DNA from ancient human remains with remarkable precision. What they're consistently finding is that the infectious disease landscape of prehistory was far more complex and crowded than anyone had previously imagined. Teeth are particularly useful because the pulp chamber inside a tooth can preserve pathogen DNA across thousands of years in ways that other tissues can't reliably manage. Researchers have pulled recognizable bacterial and viral sequences out of teeth from skeletal remains ranging from the Bronze Age all the way back to populations that predate writing, agriculture, every institution we associate with civilization. So what would an ancient human have actually experienced when they contracted one of these infections? It depended entirely on the specific pathogen and the state of that individual's immune system. That is more or less the same answer today. HSV-2 would have produced the same painful lesions it produces now, with no treatment available and no understanding of what was causing them. HPV would have gone unnoticed in most people most of the time, as it still does. In some people would have caused visible growths. Gonorrhea would have produced the discharge and discomfort that characterizes it today. In some individuals would have progressed to more serious complications. Without antibiotics, a bacterial infection that reached that stage had no reliable ceiling. The body either cleared it or it didn't. Ancient humans were not entirely without resources. It would be a mistake to picture them as completely helpless. Archaeological evidence from sites across Africa, Europe, Asia, shows that both Homo sapiens and Neanderthals were using plants medicinally long before any written record of medicine existed. Analysis of dental calculus from Neanderthal remains at El Cedron Cave in Spain identified traces of yarrow, groundsel, poplar bark, all of which contain biologically active compounds. Poplar bark in particular contains salicylates, the same chemical family that aspirin belongs to. Some of those plants also carry antimicrobial properties. Whether Neanderthals understood the mechanism is a different question entirely. What's clear is that they were selecting specific plants for specific purposes. That practice carried forward into every subsequent human culture. The oldest confirmed written medical text that describes something resembling gonorrhea is an Egyptian papyrus from around 1550 BCE, the Ebers Papyrus. That includes treatments for a urethral discharge that matches the clinical presentation closely. That document is over 3,500 years old. It addresses the condition in a matter-of-fact way that implies it had been a recognized problem long enough to have established remedies. Chinese medical texts from roughly the same period describe similar symptoms. Neither culture had any concept of bacteria, but both understood that the condition existed, that it was associated with sexual contact, that certain treatments offered some relief. The condition itself is far older than any of those texts. The texts are just the first moment someone thought to, "Oh, write it down." The question of why sexually transmitted diseases exist as a category at all is actually worth a moment of consideration. From an evolutionary standpoint, a pathogen that transmits through sex has a very specific strategy going for it. It lives in reproductive tissue. It spreads during the most intimate contact two organisms can have. It generally doesn't kill the host quickly because a dead host stops reproducing and stops transmitting. A herpes virus that causes manageable symptoms is more evolutionarily successful than one that kills within a week. Chronic tolerable infections that leave the host alive and reproductively active are exactly what natural selection would favor from the pathogen's perspective. That's not a design flaw. That's the strategy working precisely as intended, just not in anyone's favor but the virus. What that also means is that these infections weren't some anomaly in prehistoric life. They were part of the baseline biological reality of being a sexually reproducing animal in a world full of other organisms doing the same thing. Every species that reproduces sexually is a potential vehicle for sexually transmitted infections. Chimpanzees, bonobos, gorillas, and most other primates carry their own versions of these pathogens, many of which transferred to hominins at various points across millions of years. The specific diseases that ended up established in Homo sapiens populations are a function of which contacts happened, that pathogen transfer succeeded, and which organisms adapted well enough to a new host to persist across generations. We didn't inherit this particular biological situation because of anything our ancestors did wrong. We inherited it because they were alive. Ancient humans had no germ theory. They had no understanding that tiny invisible organisms were causing their symptoms. They had no concept of transmission routes in any medical sense. What they had was pattern recognition, the accumulated experience of observing which behaviors preceded which outcomes. A gradually expanding toolkit of plant-based remedies refined through trial and error across thousands of years. Whether that knowledge was ever specific enough to address sexually transmitted infections in particular is genuinely hard to know. The archaeological record of prehistoric medicine is fragmentary at best. The parts that survive tend to reflect the most durable materials rather than the most common practices. What we can say with confidence is that the people carrying these infections were not passive. They were doing something. They just had no name for what was making them sick. So yes, ancient humans got STDs. Some of those infections are still circulating today in forms that are genetically continuous with what was present in prehistoric populations. The herpes virus that has been in the primate line for 6 million years, the pubic lice that jumped from gorillas 3.3 million years ago, the gonorrhea that evolved alongside Homo sapiens from its earliest appearance, the treponema bacteria showing up in skeletons that predate the story everyone agreed on for 5 centuries, none of this began with modernity. None of it began with cities or density or any particular set of behaviors that are easy to attach to recent history. It began the moment living bodies made contact with other living bodies. That is approximately where every human story begins.